I would like to reiterate something that Jean said, and that is that this colloquy owes its life to Dr. Tomatis, but the inspiration for it came from Rich DeVos himself, who thought that the ethics of medicine were just as important as all the technological advances that we're making. And especially tonight, those come in brilliant contrast with genome editing. When I was in medical school, I didn't even know the term existed. Genome editing was fanciful fiction, so it's very special. And we're privileged to have two really good speakers. The person that was intended to be the moderator for today was Dr. MacKeigan. And Dr. MacKeigan initially joined the faculty at the Van Andel Institute, and then recently, I think it was in 2017, that he moved over to Michigan State University and works in the laboratory just down the hill from where he used to work. And he does gene editing as part of his daily activity. I should mention, before I forget, I can actually read this, that he got his bachelor's degree from the University of Colorado and then went to North Carolina for his Ph.D. And postdoctorally, he was at Harvard Medical School and then temporarily joined the staff at Novartis. It's a big pharma, I think. And so he's had quite a bit of experiences, and I think we're very lucky to have someone with his experience now that Alta Charo can't come. So without further ado, Dr. Jeff MacKeigan.
Such a privilege to be introduced by Dr. Farr. Feels like family. He knew my father quite well, and it's just a privilege to have you introduce me. My job today, before we get to the more panel session, is really to introduce a leader in the field, what we call a rock star in the world of genome editing and providing high quality care to pediatric patients. Dr. Matthew Porteus is an associate professor at Stanford University. He received both his combined MD and PhD degree from Stanford, where he focused on mammalian forebrain development. He then did his postdoctoral fellowship at Boston Children's Hospital, his residency and clinical training at Boston Children's Hospital, and his postdoctoral fellowship just down around the corner under the guidance of David Baltimore. Dr. Porteus is considered a pioneer in the field of genome editing, with numerous publications found in many high-impact journals such as Nature and the New England Journal of Medicine. He's received numerous awards and grants from the National Institutes of Health, the Burroughs Wellcome Fund, and the Howard Hughes Medical Institute. He most recently received a large award from the California Institute of Regenerative Medicine for his research on CRISPR technology and for applying that to correct a mutation in sickle cell disease. So let's get started and welcome Dr. Porteus.
All right. Well, thank you to Dr. Farr and Louise. Sorry, I can't say your last name yet. And Jeff for that really kind introduction, and thank you all for coming and spending the next half an hour listening to me talk as Matt Porteus, and then the next 20 or 25 minutes talking to you as Alta Charo. Alta is a JD, and I have a lot of letters after my name, but JD are not two of them. So I'll try to do my best impersonation of somebody who is impossible to impersonate. So let's get through that. So what I'm going to talk a lot about in this first talk is sort of the science and technology of genome editing using the CRISPR-Cas9 tool, and then end with a little discussion on some ethics of it. And then as Professor Charo will go a lot more into the regulatory and ethics of genome editing. So what is genome editing? Well, genome editing is this new way of changing the sequence of the DNA within a cell with single nucleotide or single letter precision, so that we can now alter the sequence of DNA, as I said, just like we can alter a Word document on our computer. But it's a little bit more difficult. And the way it works is that we design a protein, and there's various different types of proteins we can design, but all of them work in the same way, which is they're designed to recognize a specific sequence in the genome. And when they bind to that sequence, they'll cut the DNA into two parts. They'll make what we call a double-stranded break. And at that point, the cell wants to repair that break, wants to put the two ends of the DNA back together. And it can do it in one of two ways. One way, going down the right-hand side, is a process we call non-homologous end joining, in which the two ends are simply stitched back together. This is generally an accurate process, but if it's completely accurate, of course, it reconstitutes the sequence that the nuclease bound to, and so it recuts it again. And if you get cutting and repair, eventually you can create little nibbles at the site of the break, what we call insertions and deletions. And this allows you to create mutations at specific sites in the genome. And so this is a good way of breaking things. The other way that the cell repairs double-stranded breaks is by homologous recombination. And we can harness this homologous recombination pathway by not only creating a specific double-stranded break, but also by providing an extra sequence of DNA, what we call a donor DNA. And the homologous recombination machinery of the cell will use this donor DNA through what you can think of as a copy and paste mechanism and make a good copy of the undamaged donor DNA and paste it over the break. And by designing the donor DNA to have sequence changes, we can introduce specific sequence changes, single letter changes, or actually introduce large cassettes of genes precisely into the genome. And so this is what we mean by genome editing. And so, again, going to our laptop computers, and I don't even think anyone has real computers anymore, one can think of genome editing as conceptually as a find and replace. So you ask the genome editing machinery to find a disease-causing sequence, and then you engineer it to replace it with a non-disease-causing sequence. And so there are a number of different ways we can make that double-stranded break, and those are shown on the right-hand side here. Meganucleases, zinc finger nucleases, tau effector nucleases, and the latest tool called the CRISPR-Cas9 nucleases. And using this, as I mentioned, you can add, delete, or inactivate genes. You can, as I said, it works by using the own cellular repair mechanisms. But what has really transformed this field in the last 5 years is the discovery of this CRISPR-Cas9 platform. And that's because it's really democratized the ability of research labs to do genome editing. And the reason for this is that instead of having to design a protein that recognizes a sequence of DNA, which is very difficult to do because we don't understand the rules, instead we use a small RNA that we can design to recognize DNA based on Watson-Crick base pairing, and that brings the nuclease to the site of the break. And so it's much easier to make a CRISPR-Cas9 nuclease. It turns out that this system is highly efficient in a wide variety of cell types, including somatic stem cells and zygotes, and we'll get into that. And it's actually highly specific. It's not perfectly specific, but it's very specific. And so there's been an explosion in the use of this tool for basic research applications. Now, I am a pediatric hematologist oncologist, and I got into this field because I was interested in developing better therapies for sickle cell disease. And sickle cell disease is one of what we estimate is at least 10,000 diseases, so-called monogenic diseases, which are caused by single variants and single genes leading to specific diseases. Most of these diseases are quite rare, maybe only affecting a few hundred people around the world. But in some, these diseases affect a large number of people, both in the United States and worldwide. It's estimated that perhaps as many as 30 million people in the US have some sort of monogenic disease. And they affect all aspects of medicine, from hematology to the lungs, to the immune system, to the heart, to the skin, to the brain. And many of those diseases are listed. And then the other point I want to make is that while many of these diseases are rare, they impact on a much larger community of people. Not only their immediate family, but the schools and their friends and their extended family. So a single patient it's not just one patient, you're actually affecting a large community of people. So the echoes of this disease. So what we'd like to do then with genome editing is apply this precision to making changes in the DNA as a way of correcting these genetic diseases right at their foundation. So we can think about doing genome editing in one of two ways. So one way is what we call ex vivo genome editing. And what this would involve is, both of these are somatic cell genome editing. So let me take those terms in order. So somatic cells are cells that live within us or make us all up, but when we die, the cells die. They do not pass along their genetic information to future generations. And so we can either modify somatic cells by bringing them outside the body in an ex vivo setting, modifying them using the genome editing machinery, which I'll talk about, and then giving them back. So one can imagine using this to correct genetic diseases of the blood, like sickle cell disease, or developing better cancer immunotherapies, or a whole host of other diseases. Now, the other approach to doing somatic cell editing is to do in vivo editing, which would mean delivering the genome editing machinery into the body. And so that would allow us to, say, edit liver cells for hemophilia or diseases of the liver, or the muscle cells for muscular dystrophy, or maybe even brain cells, neurons, for Huntington's disease. So if we contrast ex vivo versus in vivo editing, there's pros and cons to both. So as I mentioned, ex vivo editing involves being able to isolate the patient's cells, modifying them outside the body, and then having a way of giving them back to the patient so that they'll take and last for the lifetime of the patient. So what's the advantages? Well, since we have the cells outside the body, it's easier to control the delivery of the genome editing machinery to those cells. It's easier to measure what we've done both where we want and where we don't want to have things happen. But the disadvantage is we have to know how to isolate these cells, and we have to know how to give them back. And for many of the diseases that I've mentioned, we don't know how to do that. For diseases of the blood and immune system, we do. So what's about in vivo editing? As I said, it's delivering the genome editing machinery to the patient to genetically modify the cells where they sit in the liver or the organ that they normally belong in. And so this gives us a potential method to edit cells for which we do not know how to isolate and expand and transplant. Cells of the eye or the brain or the liver or the somatic muscle or the heart. And it doesn't involve any cell manufacturing outside the body. What are the cons of this approach? Is that because we're delivering the drug, the genome editing drug, into the body, we don't control the on-target and off-target effects as well as we would if we were modifying the cells outside the body. And there are significant challenges to delivering the genome editing machinery to the right tissues. How do we get our machinery to the liver and not to other tissues? And then finally, I'm going to mention a little bit that there's problems that the genome editing systems we use are foreign proteins, and we have the problem, the barrier of the human immune response. So I'm going to focus in the next few slides on what we've been doing in terms of applying genome editing to sickle cell disease. So sickle cell disease is caused it's an autosomal recessive disease caused by a single nucleotide change in the beta globin gene, an A to a T, that leads to a single amino acid change in the protein from a charged glutamic acid to a polar valine. And when you have a hemoglobin molecule that's normally a tetramer in red blood cells, with the sickle cell change, it polymerizes, and it converts a round red blood cell into a stiff sickle-shaped red blood cell, giving the disease its name. These stiff sickle red blood cells can cause occlusions in the blood vessels and prevent oxygen from getting delivered to the distal tissues. If you have this disease in Africa, the median lifespan is on the order of 5 to 8 years of age. If you have this disease in the United States, the median lifespan is in the mid-40s. So it shows actually that we can modify this disease by having a reasonably good healthcare system, but we still don't achieve what we want. So fundamentally then, we want to change hemoglobin S, that GTG, to hemoglobin A, a GAG. The disease only affects red blood cells. And so red blood cells live for about 100, normal red blood cells live for about 100 days. So we don't want to modify red blood cells because we would only get a transient benefit from that. So instead of trying to modify or correct red blood cells, we have to correct hematopoietic stem cells. So hematopoietic stem cells are somatic stem cells that generate all of the blood in the body for the lifetime that we live. So if we can correct the hematopoietic stem cell, we can then get red blood cells that don't have sickle cell disease for the rest of their life. So one way of giving somebody corrected hematopoietic stem cells is by doing what we call an allogeneic hematopoietic stem cell transplant. there's a mouthful for you, or a bone marrow transplant. And in this procedure, we take bone marrow stem cells from somebody who doesn't have sickle cell disease, and we give them to somebody who does have sickle cell disease. And we create space for those stem cells by giving the patient high doses of chemotherapy to allow our stem cells to take. And it turns out that if you have an appropriate donor, this works quite well. There's about a 95% cure rate by doing a allogeneic stem cell transplant for sickle cell disease, and this was a paper published in 2016. The problem is that with this allogeneic stem cell transplant, only about 10 to 15% of patients actually have a donor. There's still a 5% mortality from the process, and there's problems because it's a immunologic mismatch. You have problems of poor immune function and graft versus host disease that limit the places and number of patients who actually can receive this therapy. So what we've been working on then is replacing allogeneic cells with the idea of correcting the patient's own cells. And we've been using the CRISPR-Cas9 tool to do that. And we use the CRISPR-Cas9 tool in the following form, which is what we call a ribonucleoprotein complex. So we take the Cas9 protein, which is depicted here in this gray blob, which has multiple functions, but the most important function of which is that it can make that break in the DNA. But in order to know where to make that break, it is guided by what we call the guide RNA, which is shown there in the little bubbles. And so the 20 nucleotide sequence that's shown with a bunch of lines there tells where this complex should go and make the break. And so we deliver this complex, we make this protein RNA complex in a test tube in the lab, and then we mix it with our cells, and we we electrocute the cells basically to make holes, and this complex gets in. So that makes the break. Now we need to deliver that piece of DNA that's going to fix the break and fix that mutation. And for that, we actually use a virus. And the reason we use a virus is that viruses have evolved to get into cells without them being detected. What we know is if you deliver DNA into cells, cells have a really good way of detecting that as a foreign invasion, so we harness the ability of viruses to get into cells. So in our process then, we deliver our nuclease as this RNP complex, and then we follow that by delivering the virus to give that correction, that donor DNA. And without going and summarizing about 4 years of work and about 5 people's 4 years of work is in this one slide. And what we're able to do now is if we take stem cells from patients with sickle cell disease, so we call those, they're marked by a protein called CD34, and we measure how many genes did we correct in those patients' cells, we find that we can now, on average, correct about 70% of the alleles in patients' hematopoietic stem and progenitor cells. You might be asking yourself, well, 70% enough? And it turns out it probably is enough. Now, we have some debates about whether 2% would be enough, or 5% would be enough, or 20% would be enough, but everyone agrees that 20% correction would probably be sufficient to cure this disease. So this is now what we're thinking about doing next year in a clinical trial, is we would isolate hematopoietic stem and progenitor cells from a patient. Now, I show a baby here because ultimately we're going to want to treat people young before the damage from the disease imparts itself, but the FDA will require us to treat older patients because of issues of consent and assent. Nonetheless, we will harvest stem cells from our patient. We'll bring them into a laboratory. We will modify them under careful what we call GMP conditions, good manufacturing practice conditions, where everything is controlled. We know basically the supply chain of everything. Those cells will then be characterized to make sure we've done what we've wanted to do and not done something that we didn't want to do. And then the patient will come back to our hospital first, but then hopefully hospitals all over the country and world, to receive those cells as part of an autologous bone marrow transplant. And that autologous transplant will require the patient to get high doses of chemotherapy, though not quite as high if they have to undergo an allogeneic transplant, to create space in the bone marrow so that our modified corrected cells can get in and engraft and start making blood. And as I said, this is something that I'm working now with it. So the laboratory team was about 3 to 5 scientists. The team now to take what we've done in the lab to the clinic is now grown to about 30 different people. It's a whole different sort of process. Now, many of you may have heard about the problem that, well, these CRISPR-Cas9 nucleases are great, but they have a lot of off-target effects. And I want to tell you that I don't think that's the biggest problem, and I'll show you why in the next few slides. So first of all, I want to put this issue of so what do we mean by off-target effects? The concern is that the nuclease, we've designed it to recognize one sequence of DNA, but it goes to another sequence of DNA and makes a break there and might cause a mutation somewhere we don't want. And we worry, are we making a mutation that might cause something bad? So let me put this all in context, though. So first of all, we have tremendous diversity in this room and in the world. So when we've done whole genome sequencing now, and I'm citing a study here that was done from Stanford, that it's estimated that we all individually contain 2.4, so our genome has 6 billion base pairs. Each of us has 2.4 million single nucleotide variants, so single nucleotide changes that's unique. So we have lots and lots of variability, and these include insertions and deletions and structural variants. So we're all born differently, fortunately. Diversity is good. But as soon as our cells start dividing, we generate even more diversity, because every time a cell divides, it generates another 10 to 20 new mutations. So mutations are sort of a fact of life. Now, eventually, we get mutations. Sometimes, well, about 50% of us develop mutations that end up leading to cancer, but that can take decades or years. And that's sort of the mutational burden that I list there. Each day that we're living, we acquire close to 10 trillion new mutations in our body. Now, they're distributed through all the cells in our body. Didn't take me quite 6 hours to fly here from California. It was about 4 hours. But just being exposed to the cosmic radiation by being in an airplane is inducing mutations in my cells. Now, about 100-fold less than just living, so I think it was worth it. Getting a body CT scan exposes people to radiation. And that generates new mutations, and so on and so forth. So I'll show you the data, but when we calculate how many new mutations might be created by genome editing on the far right, we think it's 3, 4, 5, even 6 logs lower than what's occurring in our body just by chance. So again, context here. The other thing is that the FDA has approved a genome editing trial run by a company and out of City of Hope. And they were using zinc finger nucleases, not CRISPR-Cas9 nucleases. And they found, using their nuclease that was targeting a gene that's involved in the biology of HIV, that they found 4 off-target sites. And some of these off-target sites were in the middle of genes, and the frequencies were 1%, 5%, 12%, and 20%. But the investigators and the FDA agreed that they had done a sufficient amount of functional studies to show that these changes weren't going to be harmful, and so they've allowed the trial to proceed, and it's been undergoing now for the last 3 or 4 years, and there's been no report of harm. But even as we have been developing our CRISPR-Cas9 system, Cas9 itself is being improved. And so this was work we did in collaboration with a company, and without going into too many details, they've developed a variant of Cas9 that's more specific than the variant we have been using for the last 3 years. It's about 10-fold more specific. So we can only find 2 sites where our Cas9 cuts with the wild-type version of Cas9. And now with this variant, this high fidelity or highly specific Cas9, the one site, what we call off-target 3, basically goes below the limit of detection, and off-target 1 goes from a frequency of about 14% to 1%. So we're improving the specificity all the time. So again, that's good. So invite me back in 2020. I'll tell you if our clinical trial is actually working or not. So I mentioned that the human immune system may be a potential barrier to in vivo genome editing. And why do I say that? Well, it comes from the point that the Cas9 protein comes from bacteria. The most common forms of Cas9 that we use are derived from Strep pyogenes and Staph aureus. So Strep pyogenes, strep throat, Staph aureus, which is on our skin or in our or causes abscesses or in our nose all the time. And because it's a foreign protein, when you give it to an animal, in this case, this is a study that came out of Boston, the animal will develop an immune reaction to it. So these were mice that were giving Cas9 as part of therapy for muscular dystrophy, and they cured the mice of the muscular dystrophy, but what they found is the mice developed T cells and an immune response to the Cas9 protein. Now, the mice did fine, but you can imagine that if you tried to give these mice Cas9 again, they're basically vaccinated, and you would end up with if either no effectiveness at all or an overwhelming an immune response. So we wondered that since the Cas9 comes from bacteria that are on most humans, do we have pre-existing immunity to Cas9? And what we found is that about 70% of us have, and there's two sort of flavors of Cas9 we use. One is from Staph aureus, one from Strep pyogenes, that about 70% of us have antibodies that recognize these forms of Cas9, showing that in fact our immune system already recognizes the genome editing tool that we've been working on. And this means that it's going to be challenging to give this form of Cas9 to patients. I'm going to skip this slide. So what are the implications of this? Well, one is that I think if we tried to give Cas9 to humans to modify cells in vivo, inside the body, that we could cause a potentially harmful immune response. But I think there are a lot of solutions to this potential problem, and if people are interested, we can talk about those later. Now, in the last few minutes of my talk, I'm going to give you sort of my view about genome editing of humans and the shoulds and should nots. And this idea of genome editing of humans is not conceptually a new idea. The idea of genetic engineering of humans has, of course, been around for hundreds of years, if not at least 50 years, and is the subject of books and bioethical essays and movies and so on and so forth. And it's sort of the thought process, people have divided this into this 2x2 box, where one can think about somatic cell editing to treat or prevent disease. We talk about germline or heritable editing for the treatment of disease or the prevention of the disease, somatic cell editing for enhancement, and germline and heritable editing for enhancement. And now that we have the ability to do editing so well, the question is which of these four boxes should we apply it to? And so I was fortunate enough to participate on a committee, an international study committee, sponsored by the National Academy of Sciences, National Academy of Medicine, called the Committee on Human Gene Editing, Scientific, Medical, and Ethical Considerations. And Professor Charo was actually the chair of that committee. So that's where I got to know her very well. And for those of you interested, here's a website. And actually in Professor Charo's presentation, you'll see it as well. And I'm going to go through a little bit about what the committee thought, but I'm going to take you through what this committee prompted me to think about, which is what were the important ethical principles that I would use to evaluate which potential applications of genome editing we should, which would be the ethical applications of genome editing in my own mind. And these were the 6 principles that I came up with, and actually it was very interesting for me to think through this. The first is a concept from John Rawls about the veil of ignorance. And what he's writing about here is to think about what world would you want if you didn't know where into that world you would be born. So I happen to be healthy and not have a genetic disease, but what if I didn't know that? What sort of world would I want if I might be born into this world with sickle cell disease or Huntington's disease or cystic fibrosis? I'm an MD, and maybe I'm a little TMI here. I don't know if I can really recognize what happiness is, but as an MD, I think I can recognize what suffering is. And I think we have a duty to minimize suffering. I think we also have a duty to minimize structural inequality. That doesn't mean that there will be no inequality. It just means we shouldn't create a society in which inequality is built, sort of baked into the cake. John Evans, who's a sociologist and bioethicist at UCSD, highlighted to me that we have to be careful that when we're editing people's DNA, we don't convert them into objects, that we don't treat their DNA as the individual. Instead, that we maintain people as humans. I think we also need to recognize that we need to be humble about how much we do or don't know, particularly as it relates to genetics. very simple to think, well, we understand this or that, but I think in the reality we understand very little about human genetics. We need to recognize our lack of knowledge. And finally, I think diversity is strength. And I mentioned earlier that thank God we're all different. And one of the things that kind of annoys me, not kind of, really annoys me, is when I see covers of magazines stating, human genome editing is here, or engineering humanity, and they show a picture of a blonde-haired, blue-eyed, white baby boy. I think there's a lot of implications of that picture that are, I think we should push against. So using these 6 personal principles, how do I come down on how we should use genome editing of humans? So obviously I'm working on sickle cell disease, so I think there's a lot of greens in terms of using somatic cell editing for disease. I do think there's a yellow, though, about minimizing inequality. I already mentioned that patients with sickle cell disease in the US live much longer than patients with sickle cell disease in Africa. And if we're going to develop advanced therapies, how do we make sure that these get distributed to all the people in the world who could benefit from them? I think there's many reasons why somatic cell editing for enhancement should not be done. I think they violate many of these principles. I will highlight that there is a field, a group of people out there who call themselves transhumanists, who believe that humans are a fundamentally flawed species, and that it is our ethical obligation to use every tool at our disposal to solve those flaws. I am not a transhumanist, but there are people who are out there. And I'll go into this a little bit more in the next half hour, but our committee made a little change from the past where we said under very certain specific circumstances, one might consider using heritable editing for disease. And as I said, I'll talk about that in more detail in the next half hour. And then finally, heritable editing for enhancement. It's hard to see any benefit from that. So I'm going to skip through these slides because I'll go through them with Professor Charo's talk. But I think in summary, Barry Collar, who was one of the committee members and is the chief medical officer at Rockefeller, came up with he said, we should be making healthy babies, not designer babies. And I paraphrased that to say we should be using genome editing to make healthy humans, not designer humans. And I really like that saying. So finally, what is the current status of genome editing of embryos? So in mice, and as some of you heard earlier today, it's a broadly utilized research tool. It's a fantastic way of making genetically engineered mice to study a wide variety of processes, including modeling of human disease. It's been used in non-human primates, and there have definitely been a small number of animals born, but the efficiency is quite low. So for every, for example, 100 embryos or zygotes that get injected with a genome editing machinery, only 1 or 2% of those will actually be born. And then I think there's a separate ethical issue about whether we should be engineering non-human primates to mimic human diseases because, of course, those animals will be suffering. In terms of humans, there's been sort of 3 major published studies, 2 out of China and 1 out of Oregon. And the studies out of China use tripolar zygotes. So these are in vitro fertilized embryos that have 3 nuclei in them, and so are non-viable and will quickly die and disintegrate within the first week of life. And the Chinese investigators showed that they could use the CRISPR-Cas9 tool to create genetic edits in these non-viable human zygotes, but that was inefficient, and they saw significant off-target effects. But as we mentioned, probably those off-target effects will go away. More recently, a study out of Oregon showed that they were actually trying to edit viable human zygotes for an autosomal dominant heart disease. And again, what they showed is that it remains technically inefficient. And as I'll go to in the next half hour, the disease they picked was inconsistent with the criteria that we've outlined that one might consider doing genome editing of zygotes for heritable or germline editing. And moreover, the results are actually quite scientifically controversial, and there's many people in the field who feel like what they published was an artifact. So with that, I'm going to end my half an hour. I'm going to take a sip of water and put on and try to channel Alta Charo and talk to you about some of the ethics and legal basis and regulatory aspects of genome editing. So All right. Yeah. Okay. You can do that too. Okay. So Jennifer Dowden is shown here, and Alta started with this slide because Jennifer had a quote saying, we may be nearing the beginning of the end of genetic diseases. I think that's rather optimistic. But nonetheless, there's incredible optimism about the use of genome editing. Hopefully I've conveyed why we're so optimistic. But there's also been a lot of fears about it because, as I've sort of highlighted, are we going to use it in ways that we didn't intend? And because of that, the National Academy of Sciences and National Academy of Medicine, as I said, convened an international committee to evaluate the state of the science of genome editing, the potential for clinical applications, ethical, legal, and regulatory standards. Could we develop any overarching principles for regulation? What were the prospects for international coordination? And what was great about this committee is that it was broad, not only in terms of where people were from, US and Canada and Israel and Egypt and France and England and China, I'm missing a few, and broad in expertise, lawyers, bioethicists, pediatricians, scientists, so a whole range of different people. And the way the committee functioned is it met multiple times, both in the US and overseas, and invited the broadest range of stakeholders we could imagine to come and talk to us about what they thought were the key issues in terms of using human genome editing. And out of that process came this report that's available online for free. And again, these slides are going to be available so you can find it quite easily. So there's three major applications of genome editing. The first is to use it as a basic research tool in the laboratory to work on cells and tissues. And the second are, again, what I've alluded to earlier in terms of somatic cell editing for patients or germline editing to treat or prevent disease. Now, in terms of basic research, which I didn't discuss very much before, one can imagine using genome editing of somatic cells, of germline cells, of pluripotent cells, so ES cells and iPS cells, as a way of better understanding how genes function, the differences between mouse and human, how human fertility and early embryonic development works, the links between genes and disease, the progression of disease with strong or even weak genetic components. And so the uses are not endless, but they're broad and they're important, and the tool's been adopted very broadly. But luckily, there's already many regulations into this type of research, ranging from institutional biosafety committees, which focus on laboratory safety, institutional review boards to make sure that people are doing research that's consistent with privacy and interest in terms of tissues and cells. And then there's the NIH Recombinant DNA Advisory Committee. And so there is already in the US a regulatory oversight to the use of genome editing for research purposes. Now, one says, well, what about embryos? Well, the use of embryos in research is not restricted to genome editing. People use those for other types of research, using other tools as well. And those are also regulated in different ways. So certain states prohibit the use of embryos in research. There's limitations on the amount of federal funding you can get, and there's issues of where you can get those embryos from. And there's definitely different public attitudes about working with embryos. And so Alta's advice for scientists was to make sure you're aware of your institutional policies, make sure you keep your funding streams, you understand your funding streams so you are using the right monies for embryo research. Make sure your embryos are coming from an ethically permissible source, and to have special considerations for transnational collaborations because the guidelines in one country may not be the guidelines of yours. A dramatic point of this is actually in Germany, researchers are prohibited on working on embryos, and in fact, a German citizen who works on embryos outside of Germany can still be arrested. So I'm not German. Okay, so that's for research. So again, there's a great it's a great tool. It will advance our understanding of human disease and basic mechanisms. but with a pretty well established regulatory oversight mechanism. So now what about thinking about it in terms of somatic cell gene therapy and germline gene therapy? So again, going back to somatic cell gene therapy, they will go into somatic cells, which will not be inherited in later generation, whereas germline therapy would be introducing changes into cells that could be passed along to future generations. In the U.S., germline gene therapy is currently not being attempted, but what would stop us from doing that? So in terms of somatic cell gene therapy or somatic cell editing, which again, I haven't said, but I should point out, somatic cell gene editing is really just a form of gene therapy. And because gene therapy has been around as a field for over 30 years, there's a well-developed oversight mechanism for how gene therapy needs to occur in the U.S. And again, this occurs through institutional biosafety committees, institutional review boards, the Recombinant DNA Advisory Committee, and last but certainly not least is the Food and Drug Administration. So before any somatic cell gene therapy, before any genetically manipulated cell or tissue is introduced into a human being, you have to have approval from the FDA. If you do not have approval from the FDA, it is against the law. And so they provide a bottleneck, I won't say bottleneck, they provide a point at which before I can introduce a cell that's been genetically manipulated, either in vivo or ex vivo, the FDA has to review what I've done and deemed it, the risk-benefit analysis as being worth trying. And they also control over functional approval as a drug. The one weakness that the FDA has, well, there's two weaknesses the FDA has. One is by law, they can't disclose what they've talked about with one group. So if I go in and talk to them about my genome editing procedure, they cannot disclose to another group what they talked to me about. So there's a lack of transparency there. The second thing that the FDA has, there's a bit of a weakness here, is that they're not allowed to evaluate the ethical implications of the research. They're only allowed to evaluate safety, do a risk-benefit analysis. The Recombinant DNA Advisory Committee contains ethicists. So the FDA, if they feel like they need ethical advice, will ask the Recombinant DNA Advisory Committee to review a protocol because they are legally not allowed to do that. So it's probably not right. So in terms of basic research in somatic cell therapy, well, particularly somatic cell therapy, as I said, it already falls under existing norms and regulatory regimes at the local, state, and federal levels. And from that, the FDA has made it clear that they were going to limit clinical trials or therapies to treatment and prevention of disease or disability at this time. The important thing is because this field is moving so quickly, that last bullet point, is that there's probably at this point no way to establish a single standard. There's no way to say this is safe enough for that disease, or one standard says that you need to hit this level of safety for all diseases, that every indication will have to be evaluated on its own merits. Now, perhaps in the future, and I know actually the National Institute of Standards is working on establishing a set of benchmarks that people can target, but those are not in place and probably not appropriate to put in place until we learn more. What about then enhancement? So I'm going to just skip. So enhancement can mean a lot of things. It can mean enhancing our ability to resist disease. It can perhaps mean allowing us to digest gluten, or those of us who are lactose intolerant or gluten intolerant. It could mean enhancing human traits like muscle mass or characteristics like perfect pitch or longevity or intelligence, although I point out that most of these traits we put here are not single gene. In fact, I would say all of these are not single gene traits, and it would be very challenging to think about how we'd even do it. And then the possibility of engineering non-human traits into humans. So again, what we mean by enhancement is making changes beyond ordinary human capacities. And so what are some concerns about fairness? Would this only be available to some people? Would there be pressure on people to seek out enhancements? We certainly do encourage enhancements in our society around enhancement through good nutrition, enhancement through education, enhancement through cosmetic procedures. Some people even call vaccines enhancements because they're enhancing our immune system. Now, I would argue a vaccine is not enhancing our immune system. It's being used to prevent a disease. But nonetheless, people have pointed that out. And then the other issue is what if I develop a genome editing procedure that is used to treat muscular dystrophy, but is repurposed to make muscle mass, say, of a non-muscular dystrophy patient bigger? The analogy would be erythropoietin or growth hormone were developed to treat underlying disease conditions, and now, unfortunately, they've been repurposed by athletes and others to enhance their traits. So what was the committee's recommendations on enhancement? That editing for purposes other than treatment or prevention of disease should not proceed at this time, that enhancement is unlikely to offer benefits sufficient to offset risk at this time. Now the challenge is, what if in 10 years we find genome editing is incredibly safe? Does that mean that we still shouldn't be enhancement? Now I think that we still shouldn't, but if you took risk off the table, would then that open the door to people using it for enhancement? And then the final bullet point was that the committee recommended that enhancement should not be done without extensive public input. So while I'm proud that the committee was broad and diverse, it was only 22 people. And that's clearly not a big enough group to make a decision like this. And so what many groups, including the national academies, are doing is continuing to support an ongoing discussion. So her advice to clinicians is to make sure we distinguish therapy from prevention from enhancement, that we distinguish innovative therapy from research, that we distinguish medical practice from use of an FDA-regulated cell-based product, and that there's an interplay with regenerative medicine, so using cells to regenerate organs that have not been genetically enhanced. Okay, so moving now on to heritable genome editing. So as I mentioned, this has been achieved in animals and is a powerful research tool. And it can be done in a number of different ways. One is to directly inject the genome editing machinery into a zygote. So after the sperm meets the egg, do the editing at that stage. The problem with that is that you don't get to analyze that zygote. You get what you get, and if you pull out one cell and analyze that, you don't know if that predicts what's going on at another cell. The other way that one could do heritable editing, though, is not to edit the zygote, but edit the cell that gives rise to the sperm, or edit the cell that gives rise to the egg, and then create a zygote from the edited sperm or egg. Now you could characterize the stem cell that gives rise to the egg or gives rise to the sperm, characterize it, and use that. So that has been done in mice, but has not been done in humans. So when you think about heritable editing, it's not all about embryo or zygote editing. You could do heritable editing without manipulating zygotes or embryos. Okay, so I think this is summary of what a lot of things that I said. So why would one want to do heritable genome editing for disease? So there are many of those thousands of those 10,000 genetic diseases I mentioned. It's possible that somatic cell editing just won't work. The disease might occur in utero before we can intervene. And so then John Evans would say, well, if that's the case, why is there a genetic right or is there an ethical right to having genetically related children? He didn't believe it, but other people, obviously, if you watch how people behave, they do. And so there's a small number of examples where you could imagine that the only way to treat or prevent a disease is by doing it very, very early. So because as soon as and you couldn't do it later. So I'm going to skip ahead and just say, okay, yeah. So this is what I want to go. So where am I going? What did I just do? Okay. Sorry. I lost track of myself. just in case you needed a review. Yeah, so what are the concerns, of course, is that the off-target changes or genetic changes may be inherited by the next generation, and the possible of multi-generational risk, that the real difficulty of any long-term follow-up, issues that we talked about today about who's giving consent for this. You're going to, the child is somebody who never gave consent to being genetically edited. And then the risk that we're going down a slippery slope, and that we might, if we're using this to engineer to do editing for disease, would somebody now start to do this for enhancement? So what the committee came to the conclusion is that caution is needed, but caution does not mean prohibition. And we propose that heritable editing for treatment of serious disease might be permitted, but only after a number of very specific criteria were met. First was that we need much more research to understand the risk-benefit. It needs to be done under strict oversight, and it needs to be restricted to a specific set of criteria, which are listed here. The first is that there's an absence of reasonable alternatives. So one reasonable alternative is, of course, in vitro fertilization with pre-implantation genetic diagnosis. And for many couples, that is a possibility. Now, we know in the U.S. that's not paid for. And so couples need to take the chance. They need to, take that chance. In other countries, however, IVF is paid for. In Israel, for example, every couple is allowed to have, the government pays for up to 2 children by IVF. That if you're going to initiate a clinical trial, it needs to be to prevent a serious disease or condition, that it has to be something very important. One of those diseases that I mentioned that probably couldn't be cured any other way. You need to change the DNA sequence back to a sequence that we know is healthy in humans. This is actually an incredibly restrictive criteria, because we're saying you can't just change it to what you think would be right. You have to document that in the human population, this sequence has been shown to be healthy. And that's actually more complicated than it sounds. So that's bullet point number 4. And that there has to be, of course, available preclinical and clinical data on the risks and benefits. There needs to be ongoing rigorous oversight during the clinical trials to make sure that the research participants are healthy and safe and are being ethically discussed with. There has to be plans for long-term multi-generational follow-up. There has to be maximum transparency and continued reassessment of both health and societal benefits. And here's an important point, with broad ongoing participation and input by the public. Not broad ongoing participation by a few set of scientists or a few set of this and that, but by the public in general. It can't be behind closed doors. And then there has to be reliable oversight mechanisms so that the use doesn't get applied to beyond treating or preventing a serious condition. So what are the key messages then of the overall report? Is that basic research in somatic gene therapy is valuable and adequately regulated by current mechanisms, that somatic therapy should be used only for the treatment and prevention of disease and disability, not for enhancement at this time, that heritable genome editing needs more research before it might be ready to be tried, and more public input is needed before it is considered. And if and when it is tried, it must be approached cautiously and only if it meets very strict criteria and stringent oversight. And with that, I will end, and I think we'll go to the Q&A session. Thank you very much.
Okay, did anyone, I forgot to mention, there's cards in your folders. If you have questions, please write them down. And then we have a couple event staff that'll be walking around, there they are, and picking those up. And so maybe we should start with a question while we're waiting, unless someone has one ready. Well, one thing, and I don't know if this totally pertains, but a lot of individuals talk about we are a shortage of organs.
Mm-hmm.
What, can you talk a little bit about that while we're waiting for a question?
Sure. Yeah, I mean, we know many people are dying because they need a solid transplant or a solid organ transplant, and they're not available. There is, on paper, a potential solution to this, which is to use genome editing as a way of engineering organs so you have more matches. But that's pretty far down the road. So, yeah, in theory, but I don't think it's a near-term solution to that problem.
And another one, while I'm waiting for others, is people talk about personalized medicine often.
Yeah.
So I get some form of cancer and I may react badly to some chemotherapeutic agent, but if you could test it out before I get it so I don't have all those bad side effects, where is that and what might we be looking at?
Yeah, so again, I'm not sure genome editing, I don't think we'd be able to say edit you to make you so that you have less side effects, but I think that genome editing does intersect with the world of personalized medicine in many ways. One is, of course, if you have not cancer per se, but say you have a genetic disease, if we can go in and correct that variant that's causing you to have the disease, that is highly personalized medicine. I think the other application of genome editing to cancer therapeutics is that we know, for example, that each cancer has antigens that could make that cancer susceptible to immune system attack. And I think genome editing is likely to play a role in enhancing our immune system in terms of eliminating cancers. Probably not editing of the cancer itself, editing of the immune system to enhance its activity, to potentiate its activity in killing that cancer.
Good, we have
Do you
one
want to add
question.
anything, Jeff?
Oh,
No,
go ahead.
Just my perspective on those sort of questions on personalized medicine. Personalized medicine, I think the definition is evolving as the years go by, and I think we're going to look back and see something like genome editing of your own immune cells to be a form of personalized medicine versus just sequencing your tumor DNA right now and trying to match a therapy to that. So that would be that I do think that it'll be a form of personalized medicine or customized T cells, if you will, that are put back into the body to attack those. My perspective also is right out of undergrad, I worked for a gene therapy company in San Francisco as my first job out of college. And so that was sort of mentioned earlier, that so the regulatory pieces that are in place have been around for certainly 20 to 30 years with gene therapy. And back to your organ question or grafting question of new organs, targeting is very difficult, and it has been for all these. And so I really see the promise being in the area of ex vivo delivery, because safe and effective and complete delivery to even a solid organ would be very difficult, obviously.
Okay, we have a couple questions here. You mentioned improving specificity. How specific does CRISPR need to be before going to clinical trial?
Well, I think the current versions are specific enough. I think what we've learned, there's a limit. So one of the other aspects of my research group is to try to actually engineer cancer in human cells and then put it into a mouse. And it's really difficult to do. So what we've shown is that when we engineer our human cells and we put them into a mice, we never see any evidence of a cell gone awry. And so I think what that means is that we'll never be able to prove it's absolutely safe using mouse models. And instead, I think the biochemistry and our analysis of where the off-target breaks suggests for serious diseases that it is now worth trying this in patients who have a serious disease in which they have no other, there's an unmet medical need. If those turn out to be, and then we have to follow those patients, and if it turns out to be safe then you start expanding the list of diseases. So this is why, again, we don't want to be using genome editing for things that aren't serious at this point, because we're only going to understand the true risks when we try this in patients.
I don't think, at least as a research tool, We use human cancer cell lines, and we'll modify those drivers of cancer called oncogenes, and we'll modify the tumor suppressors with CRISPR-Cas technology simply as a basic research tool to make one scientific change or genetic change to those cells, and then they're paired. And then we can start to work down very basic questions on how those cancer cells respond to different therapies. And my feeling, and it was discussed earlier in the afternoon session, is that the old approaches of viral integration were certainly multiple copies, upwards of 9 or even hundreds of copies of viral inserts were being placed at total random, and you couldn't predict this. As was mentioned earlier, that we can predict the insertion spots probably pretty effectively. And just daily living was given as a perfect example. Flying here was more dangerous than probably doing a CRISPR-Cas9 experiment.
How would you compare or contrast genome editing for enhancement versus cosmetic surgery for enhancement?
Yeah, right to it, you guys. That is, that is sort of the sticky wicket that everyone's struggling. What is fundamentally different about somebody having a surgical procedure for cosmetic trait versus using a genetic tool for enhancement? Is there something ethically different between those things? I don't like this answer, but to me, there's an ick factor that's different, but I can't put my finger on it. But what maybe is more concrete is that almost every surgical procedure for enhancement is reversible. And right now, if you did genome editing of cells, we really wouldn't know how to reverse that quite that easily. Now, one could imagine some reversible systems, but it wouldn't be. So perhaps it's the reversibility that allows you to distinguish the two. But it is the one that people bring up, like, well, if you allow enhancement here, why wouldn't you allow enhancement there? I wish Alta was here; she'd have a better answer.
I don't think to add.
Okay.
I would be interested if anyone else has what in their minds
this
distinguishes
the public input.
Yeah.
Isn't there a substantial difference in microphone?
Right there. Can you say your name, please?
I'm Jeff Burns. The question was about cosmetic.
Why is cosmetics?
Yeah.
Why do we do millions of cosmetic surgeries for enhancement in the U.S., and yet we would say genetic enhancement using genome editing should not be allowed?
So I wonder if that was the question. I think I probably heard the question slightly differently, which is if we do genetic enhancement, why wouldn't we allow genetic enhancement for cosmetic reasons? It seems to me that there's a substantial difference in the value there, driving people toward health versus driving people toward appearing in the way that they want or appearing in a way that seems more socially acceptable. There seems to be a substantial difference in how we value those two different parts.
Yeah.
And at least we'd want to mark a distinction like that.
Okay.
Okay. The next question, isn't it a worry that my editing treatment could overcorrect my problem? In other words, do we know what the correct DNA should be?
For many diseases, we do. So, for example, for sickle cell disease, I know the nucleotide sequence that is hemoglobin A, and we know that that's fine. And I know the nucleotide sequence for hemoglobin S, which isn't fine. And if I convert the S sequence to the A sequence, we know it's fine. And I think for many genetic diseases, that's sort of the appeal of genome editing, is you can revert a disease-causing sequence to a known sequence that doesn't cause disease. Now, people are thinking about using genome editing to engineer cells to secrete more of an enzyme to correct an underlying enzyme defect. Or gene therapy is being used to treat hemophilia, which is a disease where people don't make enough clotting factor. How do we know? We know we need to get to 10% level to have an effect. Would it be dangerous to get to 200%? And that range of that therapeutic range of efficacy and safety, I think has to come from human genetics, where we know there are people out there who don't have a problem with 150% level, and so therefore that gives us an upper limit of normal.
Yeah, just I guess two other examples of that. Certainly with a disease I study called tuberous sclerosis complex, where children are born. It's not that rare of a disease, but 1 in 6,000 incidents. They're born with a defective gene in either a gene called TSC1 or TSC2, and it is somewhat hopeful for those families to think that you could go in and repair that gene and they wouldn't have a multi-system tumor syndrome or neurologic complications such as daily seizures. But with the same example there, we know the normal sequence. There is some variation, but it still drives the normal sequence of the normal protein and its normal function. So I guess on that second case of an example of we know the sequence and we could repair it. The other comment, I've lost my train of thought on the second one, but what was there?
So just about, do you get the level right? And the other thing,
Go ahead. So I was thinking of an example from Parkinson's disease. So the PARK1 gene or the PARK1 loci encodes a gene called alpha-synuclein. And some recent studies not too long ago, about 15 years ago, showed that just gene dosage, so 5 extra copies of that gene is more than enough to drive disease symptoms and even disease in animals and in patients. And so even animal models that we use in the laboratory to study Parkinson's disease, we'll put in multiple copies of the wild type normal gene. Some people use other sophisticated techniques where we put in misfolded proteins, but highlighting that 5 extra copies is enough to drive disease in that situation.
yeah, so that leads to the level we want to achieve can be also studied in animal models, recognizing that a mouse is not a human and the limitations.
Okay, how do we define the limits of disease and disability? For example, is it ethical to use this technology to treat disabilities like Down syndrome? Is there a mechanism in place for advocating what constitutes disease and what should be left alone as diversity?
Yeah, you guys are spot on.
Yeah.
These are all exactly the hard questions that the committee wrestled with over and over again. And if you look at the report, there are gaps in the committee report because we didn't have great answers. But here's a little, some ideas around that idea. Down syndrome is an interesting one. The example that comes up a lot is the issue of congenital deafness, where there are some people who have congenital deafness who see their trait as a disease, and they would make sure to have kids that didn't have congenital deafness. There are other people who see people with congenital deafness who see congenital deafness as a range of normal, and that this is just a spectrum of human normalness, and we should maintain it. And so in fact, there's sort of these anecdotes of parents intentionally having kids who will be deaf to maintain that diversity. So I don't think it is up to us to tell any community or any parent what they should or shouldn't do. And I think one of the concerns is if we made it as if you didn't use this technology, you were somehow a less parent, that would be wrong. On the other hand, I think for serious diseases, we can develop the therapies and allow families and people to make the choice for themselves about whether they choose to use it or not. How do we define whether something's a disease or a disability or a trait? I mean, we were talking this afternoon about living in the world of gray. That's a huge world of gray, and certainly I don't want to be the one defining it, and I don't think I want a expert committee defining that. I think that's a complicated issue. I don't know. Again, I asked Jeff, and I'd ask you guys how you define that. Yeah.
Just more public input, I guess, on this is that pre-describing those conditions and when to treat and not to treat. I don't know if there's many in the audience that would agree with that.
Yeah.
Okay. Please speak to the role of, I think it says USA Healthcare, chaos funding with genomic editing.
Okay, that's a great question because I'm going to answer it any way I want. I don't quite under. So a few months ago, so there's let me go. a few months ago, I had the opportunity to testify in front of the Senate Help Committee. Lamar Alexander and Patty Murray chair that committee, and they were very interested in the technology of genome editing. And they were very nice, and they treated us, the three panelists, quite respectfully, and they were highly interested in what we were doing, and they had words of praise. And so that was great, and this is a new technology. At the same time they were having that committee meeting, the renewal of CHIP, the Children's Health research funding for children who didn't have health research, hadn't been reauthorized. Now it's been since reauthorized. And I agree that there's a little disconnect here between being excited about future technologies without providing the funding and insurance mechanisms to take care of people today. And that we should all be fighting for in our own time. Hopefully I answered the question that the person tried to ask, but
Maybe
there is a disconnect here.
What's the projected cost for repairing sickle cell disease, and maybe just shock us a little bit?
Yeah, so there's let's see, I think there's 3 gene therapy drugs that have been commercially approved, 1 in Europe, 2 in the US, and the cost for a single dose of the therapy is ranging between $300,000 and $800,000. That's not including all the ancillary costs of taking care of the patients. That's just for the drug itself. Now, some of those costs are, now for sickle cell disease or hemophilia, where the lifetime cost might approach $10 to $20 million, one could say a one-shot curative therapy at $500,000 is cost effective. But for other diseases, that calculus is less well established. Yeah.
Okay, Dr. Porteus, in your interactions with patients with sickle cell disease, have they shared their thoughts on the potential of gene editing?
So I've had I've been able to talk to families in the Palo Alto area about genome editing, but I don't think that that's a representative community of sickle cell patients. Clearly, I'm getting a biased sample. They're very interested. They're excited. They often admit that they don't know a lot. But it is also a community where I've interacted with some of the leaders of the sickle cell communities, and they admit that their community, well, I wouldn't say admit, they acknowledge that the community is undereducated. And one of, I think, my New Year's resolutions for 2018 is to become more actively engaged in those communities and make sure that not only are we working hard in the lab to develop this, but also making sure to engage the community and educate the community directly and listen to them.
Okay, the next one. What are the solutions to using CRISPR-Cas9 in individuals who already have antibodies to Cas9?
Yeah.
How can you tell who has the antibodies?
Yeah.
And will the use of other Cas nucleuses prevent this issue?
Yeah, so I think that detecting whether somebody has pre-existing immunity is pretty simple. We're not a really sophisticated immunology lab, and we could do it. So I think that will be pretty easy to do with a blood spot test. So what are some of the solutions? I think the person who wrote the question implied probably the most likely one, which is to develop Cas9 systems from bacteria that don't infect humans. We know these Cas systems exist in millions of different bacteria, and so it's just a matter of people now going into that and developing some system from bacteria that don't infect us. Another intermediate solution, because the Staph aureus and Strep pyogenes Cas9s are so well developed, is our study just looked at adults, and it's possible that, for example, young children or infants haven't yet developed immunity to Cas9, and therefore there would be a window of opportunity in terms of using in vivo genome editing before they develop that immunity. And for many, serious genetic diseases that infect infants, that's not still a path forward. So I think we need to understand when in life this immunity develops.
And just a question for you: do you have any autoantibodies against when you do it ex vivo? So obviously you're implying a lot of this was an application that might be a little farther away in vivo. But what about the ex vivo reactions?
So by the time we give the cells back, the Cas9 is gone. So we don't think it'll be a problem for ex vivo problems, but we'll see.
It's degraded and turned
It's
over
degraded.
in that time. Yeah,
that's right.
Okay, enhancement therapy is used, example, growth hormone for short stature.
Yeah.
Do any of the criteria used for this apply to other enhancements?
Yeah, so, and short stature is another great question. Is that a disease or a trait or disability? Now, clearly, parents feel like they want medical therapy, and so they get growth hormone for that. And what, how, there are pediatric endocrinologists in the room, what is short? It's a very complicated question. The other thing, as I'm talking about this, the use of growth hormone is one of those, the prime example about repurposing a drug. So it gets approved for one indication and it gets used for another. The FDA actually has put restrictions on the ability of physicians to prescribe growth hormone. You can't prescribe growth hormone unless you have registered with the FDA to prescribe growth hormone. But they don't like that at all. They do not feel like that's an effective system to prevent the broad use of growth hormone. I just said a lot without answering a question because I don't think I have a good answer. Yeah, Jeff.
No,
Okay, I'll give another one then.
Yeah.
What are current projected costs to patients, such as one with sickle cell to proven therapy? I think this was answered. Any current coverage by insurances
scooped
or
that
private
one.
paper experimental, I think was the other piece of it, covered in a trial or something.
Yeah. So how do it's a more the general issue is how do patients who are enrolled on a new clinical trial, how is that paid for? So if they're eligible for certain, so standard of care costs will be paid for by insurance. The experimental therapy then has to be paid for by someone, and that's sometimes a really challenging issue is who's going to pay for that. Hospitals don't like to cover that. So that's why you often are partnering with biotech or pharma so that they pay for the clinical costs of the drug. But then or you can get philanthropy to cover it. But yeah, so usually in these clinical trials, the costs of coverage is a mixture between insurance because you're giving standard of care therapies. For example, sickle cell disease need blood counts periodically. So we don't have to pay. We're not going to have to pay for those blood counts, but we'll have to somehow pay for the drug product. So it's a hybrid.
Yeah, so the clinical development of any
Yeah.
drug or biologic in this instance needs a commercialization path. And so to have a clear commercialization path that usually, unfortunately, always involves industry and at times, rarely, more often, philanthropy. And then the other thing is, which has been discussed already today a little bit, is the regulatory path has to be pretty clear. And without those two things, it's almost impossible to get a clinical trial run in this country.
Now there's two questions very similar, so I'll kind of try to put them together. Would the cost of this procedure allow a line between those who can afford it and those who cannot? Would it make those in poverty more susceptible to diseases or not getting the appropriate treatments? And the second one is very similar.
All right, so I'm going to raise my pediatrician flag. Yay for universal health care, right? If we had universal health care, that question would not be relevant. So I'm gonna stand by that answer. But I will go back to the issue of that's for citizens that's for people in the U.S. Or in people who live in countries with good healthcare systems. How do we get this to patients? So most patients with sickle cell disease live in Africa and India. How are we going to get this? How are we going to get the process streamlined enough so it can be delivered to people in countries where healthcare resources are minimal? And how do we do it in a way that's cost effective? So I think questions to be solved. I think they're going to be questions that will be answered by people who are in high school and college right now.
Okay, this one is bone marrow transplant programs have been limited by certificate of need programs, resulting in financial gain for some institutions. Should financial profit be a factor in democratizing the gene editing world?
Yeah, it's another spot on question, and it goes to the I guess to use from the Belmont criteria, the issue of justice, and should we only be bringing therapies to market that can make a company a profit? And I hope not. In fact, I really hope not, because we've already seen, well, let me step back a little bit. The gene therapy drug that's been approved in Europe for the treatment of a form of bubble boy disease called ADA SCID was licensed by GlaxoSmithKline, GSK. And they helped develop the product, and they took it through registration, and they can sell it they can deliver it sell it as a drug in Europe. They've backed out and said, we don't think that this will make us any money, and so we're not going to sell it anymore. And so they're looking to sort of spin out a company that will deliver it. There may be a new model. Maybe I'm just too idealistic, that we're going to have drug companies that deliver drugs at cost without looking to have a profit. Who's going to do that? I'm not sure. But it also gets to the point that of those 10,000 genetic diseases, sickle cell disease, thalassemia, certain others are common enough that their biotech pharma are going to be interested because they're going to be able to make money. But 9,900 of them don't fall into that category. And I think we have to find a solution to be able to develop curative therapies for diseases that might only affect 10 or 20 people a year around the world. We don't have that model right now in the healthcare system.
With genetic counseling, in the future, could couples have genetic changes made in egg sperm to prevent hereditary diseases, high lipids, Parkinson's, Alzheimer's, diseases of aging?
So, yeah.
In one,
I'm going to be brain dead. No, these are great. These are absolutely great questions. So a lot in that question. So first of all, most cases of Alzheimer's are not genetic. Most cases of Parkinson's are not genetic. So most neurodegenerative diseases are not genetic. So but that being said, a small fraction are. And so then could we use genome editing to prevent somebody from developing Alzheimer's by editing, sperm or egg to create embryos who would not be at risk of developing genetic Alzheimer's, genetic Parkinson's, or genetic neurodegenerative diseases? I think first we have to show, and that's really complicated, right? Because you're doing something before birth to treat a disease that's going to show up 50, 60, 70, 80 years later. I think we need to know a lot more about, about the safety of genome editing before you'd want to take on something like that. And you'd want to make sure, again, the first criteria is there's no other reasonable alternative. One of the things I forgot to mention is that if we got really good at somatic cell editing, we wouldn't have to do germline editing. So for example, if one could figure out how to do in vivo editing of neurons to take care of the genetic synuclein mutation or a Parkin mutation, then we wouldn't have to go back to germline editing. So the better we get at somatic cell editing, the less need or less drive there will be for germline editing.
Yeah, and I think it also raises the point back to gene therapy is that effective targeting and delivery vehicles of these technology is really going to be the limitation, is what we all see now in front of us, is that the specificity and the precision and the safety is there. It's now we need delivery payload, and that's been around for 30 years that we haven't found a way to effectively deliver these gene therapy approaches.
At what point does a treatment fall into enhancements? For example, would a trans individual attempting sex change be considered enhancement?
I don't know. Yeah, I don't think so. I think that's realizing one's potential, but I don't think that's enhancement.
Okay, does gene editing, or can it affect the epigenome?
So, yeah, what I didn't talk about today. So certainly the process of gene editing, making a break in a gene, does change the epigenetics surrounding that break. It's thought that that's a transient change and will be reversed once the break is repaired and everything goes back to normal. But that truly is a question that needs to be studied further. And certainly if you're modifying a gene and having that cell behave differently may have epigenetic effects throughout the genome that we don't understand. So certainly an area of interest. But the other thing that we didn't discuss is people have talked about epigenome editing, so which is sort of a weird term, but it's a hybrid, which is not to use the Cas9 protein as a way of making a break in the DNA and changing the DNA sequence, but instead using the Cas9 system to bring an epigenetic modifier to a certain site and change the epigenetics around a certain site, and change the way that gene is expressed or how long it's expressed or to what level it's expressed. It wouldn't be the same as genome editing in the sense of changing the DNA sequence, but it would be a way of changing the way a cell functions without having to change the DNA sequence of a cell. And there's a lot of research about how we would do that. In terms of translational applications, it's a lot farther behind because it's unclear exactly when or how you would use that. I don't know, Jeff.
No, I think we talked about it in the afternoon session about this Cas9, which the D stands for dead, and it doesn't have the enzymatic activity to cut the DNA. And you can use that enzyme along with the guide RNA to bring what we'd call payload, or that could modify both marks on DNA or modify marks on histones that would affect gene expression versus DNA sequence. So I agree that I think that is could be a very valuable research tool, but I don't see the clinical application anytime soon.
In the field of genomics, is the field of genomics progressing at an acceptable rate? If not, what is the greatest constraints? Is it trained scientists, lack of equipment, lagging knowledge, inability to accelerate all of the data, examine all the data generated?
I guess I'll take that one.
Yeah,
first. I'll
you
be the
can
first
take
one to
that
take
one.
first. No, I guess one comment would be is with what we, I mean, it was sort of emphasized earlier also that what we can do now for the cost of what we can do is just mind-boggling with the drop in prices of, say, genome sequencing to approach just over $1,000 for a genome, where 20 years ago it was a $100 million operation. So now in our laboratory, to give an example, is we looked at the rare disease tuberous sclerosis, and within about a 2.5 year period, we were able to provide more information, sequencing data on those disease populations than was collected prior. And so that was one of the things, the barriers was active patients that want to participate in rare disease. They're all usually all very active and want to participate, but finding patients in a rare disease is difficult. So you have to have a sort of a national search for those patients. And the second thing is money. It turns out that if you want to get politically active, is that our National Institutes of Health provides a wealth of resources for biomedical research in this country, but with cost of research equipment, we have an annual drop of a few percent every year in how far research dollars will go. So not only now is research quite exciting in what we can discover and maybe translate, the competition for dollars is very high. And so an example would be we're funded by the National Cancer Institutes, and the pay line, which is who gets funded and who doesn't, is approaching about 8%. That means 92% of people that are very talented, that have great ideas, that work their whole careers and lives for this, and the people they have in their labs and employ cannot compete for funding successfully. So basically 1 in 10 researchers out there gets federal funding right now.
And I agree exactly. The other thing I would add is going back to the earlier question about developing therapies for rare diseases. We need to train and fund more experts, fund translational medicine and people trained in translational medicine. Mm-hmm. So the NIH has historically been, we need more money, I get all that, has historically been good at funding discoveries. And biotech and pharma have been pretty good, we can, not perfect, at creating drugs for common diseases. But what we've been missed is that what we need now, particularly with, I think, genome editing, is a large cohort of people who are trained and ambitious and driven to using these new technologies to bring therapies to rare diseases. Because, as I said, I think, we can affect the lives of not only those patients, but large communities but we're missing that. It's hard to get trained in that right now.
Okay, speaking to minimizing inequality in genome editing, how do you feel these gaps can be closed or reduced in the future of genome editing?
Well, I'd probably get stuff thrown at me. I mean, one way is for to go from to start taking the technology and moving it into places like China and India and Brazil, which are, second world. I don't even hate that term, but let's just say, not maybe where we're at, but still a lot of smart, sophisticated people with a lot of patience. And, they can make generic drugs pretty cheap. Maybe they can take the process that we're doing in a very expensive fashion in the U.S. and find ways of doing it much more cost effectively. So it may be one of the ways of solving this problem of distribution is to, get a medical center in India to adopt this, and a medical center in Brazil, and a medical center in China to adopt it, and they will figure out ways of making it work for their communities.
And the last question before we do open mic is basically very similar to the earlier one. Do you perceive gene editing techniques and technology becoming trademarked or commercialized to an extent where possible to sell the opportunity for personal and cosmetic pieces? Do you see that coming soon?
I think I know, through my interactions with the biotech firms that are, founded around genome editing, and the pharmaceutical firms founded around, they don't want to come near that right now. So I don't see it coming soon. But I think the question is if in 10 years there's 100 clinical trials showing that this is all safe, will someone say, now I'm going to start a cosmetic company based on genome editing? I don't know.
Okay, now is the opportunity. If you want to have a question from the mic, raise your hand and someone will come to you. Do we have any additional questions, or are we?
There's one here.
There's one over at John.
I'll just shut up until the mic comes. I wanted to thank you for coming. First of all, I thought what you did was fantastic, and from my perspective, the way you explained it made great sense to me, and I really, for all I've heard about genomic editing, it just flies over my head, and I have a much better grasp on that. So I just wanted to thank you for that. Thanks. And for doing double duty also. And Jeff, thanks for pinch hitting as well. So the question I had was, when we think about things like Huntington's and sickle, boy, it's really specific. We know how to do that. And I'm anxious to see those, the results of the fruits of that labor coming forth. But I was sitting here thinking about mental health. Mm-hmm. And thinking about alcoholism, thinking about opioid addiction, and some of the things that really ravage our country right now. Many people have speculated, and some have demonstrated, that there is, in fact, some element of genetic underlying hiccup that results in a predisposition for addiction. Mm-hmm. Is that something that we're even beginning to broach into, or are we looking at it just as a really clearly physically related defects that are that you're looking at now?
Yeah, I mean, I love genome editing. I spend way too much time thinking about it, but I don't think it's, a magic wand that's going to solve all the problems that we have, like there's many of which you mentioned. Are there perhaps potential strategies that can be utilized for these diseases in the future? Perhaps. But I think if I came up here and told you that genome editing was the solution to everything, you should probably throw me out. That's just not going to be happening. I think you've highlighted some things, and maybe it goes back to the earlier point, is that I love that, this Lamar Senator Alexander is excited about genome editing. I wish he was more excited about putting in all the hard work that needs to be done to solve the problems that you were mentioning. And I wish there was an equal excitement about tackling those problems as there are to figuring out what the next great variant of Cas9 is. Unfortunately, we have a mismatch here.
I think you talk about, and you brought it up earlier, polygenic diseases and environment interactions versus a monogenic correction of a disease gene. Certainly, maybe a question for you, Matthew. How many genes do you think you could repair in one setting? Is it 8? Is it 6? Is it? Yeah. Am I way off?
So right now we're up to 4.
Okay, so I was a little bullish.
But only a small percentage of cells get
Yeah. I'll get all 4, yes.
to get all 4.
Yeah.
So, some get 1, some get 2, some get 3, some get 4. And the other problem also is the more genes you try to modify at once, the more genetic chaos you create as well. Because, like I mentioned, what I didn't mention is that if you make 2 breaks in the genome at the same time, at some frequency, the cell won't put the 2 ends back together where they're supposed to. It'll put the 2 ends, it'll swap them. And so you'll create chromosomal translocations and chromosomal deletions. So the more things you do at once, the more sort of genetic chaos you might create. And so then that becomes, I think, a limiting factor in terms of going after polygenic diseases. I also think that, we don't, my point about humility is we don't understand that, polygenic variants that might predispose you to a condition that exists in 21st century America may be a healthy combination of traits in a different environment. And for us to think that this set of traits is always pathologic would be, I think, overestimating our knowledge of what these variants that have minor effects in combination actually do.
Are there any other questions for us? Dr. Tomatis?
Dr. Porteus, you very briefly mentioned universal health care. Could you be so kind to expand in what is your thought about?
Oh, I knew I shouldn't have mentioned that. It really goes, again, it comes from the, my background as a pediatrician and my interest in developing therapies for rare diseases, and the concept of justice, that if we're going to develop therapies, they shouldn't be available to somebody just because they happen to have be employed by Facebook in Palo Alto, California, and they aren't, and instead of being in Oakland, California, where they're unemployed. I think we need to, and the only way I can think of reliably doing that is if there's some sort of universal health care. How to implement that in this country, I'm staying out of that entirely, but that's my pediatrician speaking.
Other questions? There's 2, I see. One up front and one in.
Hopefully as a comical relief for you, how insane did you go when you saw the gene theory from Jurassic Park?
I haven't seen the movies, so I haven't gone. Yeah, so I don't know.
Are we going back to the first Jurassic Park here, or because I'm also, as you're starting to figure, scientists don't watch a lot of movies. We don't have a lot of time for that, but we talked about earlier. Gattaca was a great movie. When I lived in San Francisco, that's what was playing at the theater.
Yeah.
And certainly Jurassic Park. I mean, it helps your imagination. I think for kids, it sparks maybe the Jurassic Park interest in being a scientist. Maybe scared too at times, but with that movie. But
my caveat to that, though, is again is this concept of, or I think I don't like how sometimes genetics is destiny. I think genetics is probability, it's not destiny. And I think sometimes we as scientists and we allow the popular press to portray genetic determinism. If you have this, then you're going to have that. Now, for some of the diseases we've talked about today, Huntington's disease, sickle cell disease, yeah, it's nearly 100%. But most of the things that we think of us, that we think give us our humanness, are probabilistic. And so I would love if we could get away from thinking about gene destiny, tight gene destiny relationships.
I have a question actually about one of the images you had. You had a graph that showed the exons and introns and how when you had the gene therapy put in, it didn't change too much. It didn't produce mutations. Just kind of showing that, implying anyway, that the resulting protein would still have the same activity. It's just that the percentage for the exons was very, very small, or introns, excuse me. well, they're changing between the percentages, and I'm wondering if those were the same proteins throughout, if those were the means of like accumulation, all of them.
You know what? Maybe after we're done, you can
Yes,
point me and
that's
I can
fine.
explain what that means because I'm not exactly sure which picture
the
you're talking about.
Yeah.
And look at the graph.
Yeah, we can look at the graph and yeah.
Any other questions? Seeing none, I want to thank you all for